welcome to the Beckman Institute and the second annual Beckman-Brown Lecture. I'm really pleased to introduce in one moment Dr Kiessling. But before I do that, I thought I would open by just saying a few remarks about Ted Brown, whose name is obviously associated with this lecture series. We're fortunate to have Ted here today, and in just a minute I'll ask him to come up and invite him to the stage. But Ted I would describe as a scholar first rate, someone who we all want to aspire to be like. Ted has had a prominent career here as a chemistry faculty member. He was on the faculty from, I believe, 58 until 94, and during that time served a number of really important roles. In chemistry, Ted was known for alkyl lithium reagents, nuclear quadrupole resonance, chemical kinetics, mechanism, metal carbonyl chemistry, an amazing breadth of chemistry, not to mention the author of a textbook that has been through about 14 editions. Maybe do I have the number right? And the title of it, has really come to resonate, I think, very strongly with chemists. It's Chemistry, the Central Science, and so that wasn't all that Ted did, though. He was vice chancellor for research, but then as he served in that role, recognized well before anyone else, I think, the opportunity to really move science in an interesting way. And that was really the ideas that nucleated around the building that we're in today. And it was Ted establishing a relationship with the Beckman Foundation that ultimately helped to land the funding that provided the infrastructure for the Beckman Institute. So so Ted knows our campus well. He knows, obviously, the Beckman Institute well. He is very familiar with the Beckman Foundation. And in fact, I think he has a connection with our Beckman-Brown lecturer, Dr Kiessling. And so, Ted, would you come up and say just a few words? Thank you.
Well, thank you, Jeff. Your lead-in gave me a sort of a lead-in for my remarks. You mentioned that it was my connection with the Beckman Foundation that led us to eventually receive a grant for this building from them, but it wasn't really the foundation. Well, it was. The foundation at that time consisted of Arnold and Mabel Beckman sitting in their living room with a stack about that high of proposals because they had announced that they were going to give away their money to all of it that they had, about $600 million or something like that. This is back in '78. Anyway, we got to know the Beckmans very well, and I'm very grateful for that relationship. So this endowment that we have that brings Laura here today, and that also provides for postdoctoral people, I think is emblematic of not only my professional relationship with Arnold and Mabel and the foundation, but also my personal relationship with both of them. So here we are in this beautiful building, owes its existence to the generosity and foresight of that wonderful pair of people. And we have an event which is made possible by an endowment from the Beckman Foundation, which they funded. And at one more layer, as Jeff intimated, our speaker, Dr Kiessling, was in 1994 a recipient of a Beckman Young Investigator Award, which was, the program started in '89. I was on the board of the foundation at the time. I was on the board of the foundation for 14 years. I'd come on just about that time. And this program provided sizable grants at that time to young investigators in their first tenure track position. And it was a hotly competitive business. Laura was the recipient of one of those Beckman Young Investigator Awards in '94. So she also probably has a connection through spending some time in one of those Beckman Institutes out there. I think it's called Caltech. Yeah, they have one of these things too. And she spent some time there. So all of these connections kind of make you feel that there's a sort of Beckman family out there in the world, someplace. We're all connected to somebody else. So it's a wonderful pleasure, Laura, to have you here.
Thank you, Ted. Let me offer my thanks both to you and to the foundation for making really this event and the postdocs that are possible through that endowment possible. It's with great pleasure that I introduce Dr Kiessling. She is our second Beckman-Brown Lecturer. She's currently the Novartis Professor at Massachusetts Institute of Technology, where she earned her Bachelor's in Science and then moved on and earned a PhD at Yale, followed by a postdoc at Caltech. After being at Caltech, Laura moved to the University of Wisconsin at Madison in 1991 and began a very successful career being recognized in a number of ways while she moved through the ranks. She was in 1999 designated as one of that year's class of the MacArthur Foundation Fellows, and that really is the beginning of the long list of accolades that everyone aspires to, from being a fellow of the American Association for the Advancement of Science, American Chemical Society Fellow, and then members of all of the national academies, the National Academy of Science, the American Academy of Arts and Sciences, and to name just a few. But another critical thing, if you get a chance to talk to Laura, she'll tell you about how her interdisciplinary science had played a critical role, I think, in founding what is maybe one of the most forthcoming and innovative of the American Chemical Society journal. She was the inaugural editor-in-chief of ACS Chemical Biology, and I think in many ways really moved the field of what had been an enterprise, the ACS publications, that needed some innovative thinking, and I think you've really brought that to the journal. Laura has won, as you might expect, a number of awards, and I will once again touch on the Beckman Young Investigator Awards, because I actually went and looked up what was the title of your proposal. I do. It's quite amazing. The title of her proposal was New Materials to Explore Oligosaccharide Protein Interactions. You picked an amazing topic as an assistant professor to go after, and it reflects the fact that it has been, your career unfolded now in the talk that we're going to hear today. So, just to capture in a couple of sentences, Laura's research. Her research interests focus on elucidating and exploring the mechanisms of cell surface recognition, which is, I think, what we're going to hear about today. And she's involved in really fundamental understanding of protein-glycan interactions. Her group is also interested in multivalency and its role in recognition and signal transduction. Her research covers a broad spectrum of activities, from organic synthesis to polymer chemistry, structural biology, and molecular and cell biology. And so with that, Laura, I welcome you to the podium, and thanks so much for being here today.
Thanks for that really kind introduction, and thanks for the opportunity to come here and talk about our work. And I'll say that I met Arnold Beckman when I was at Caltech. I think when I first arrived there, they dedicated the Beckman Center, and it was one of the first events that he went to after Mabel had died. And he gave this incredibly eloquent speech about, the importance of science, intertwining it with technology. And then he got to the part where he wished Mabel had been there with him. And I just remember thinking, I hope that someday I will meet someone that I can love for like this long a time. Like, he was such an amazing scientist, and I don't know, you got a glimpse of him as a person, not just as a figure or an entrepreneur, but really as a person. So thank you for reminding me of that. And I will also say that when I was a Beckman Young Investigator, we had to give talks in front of the selection committee, and these eminent scientists were terrifying to us. I mean, and then after I gave that talk, they all were so generous and kind. It was like, oh, okay, I didn't need to be terrified for the last whatever it was. So anyway, I'm going to tell you guys today about some work that my lab has really gotten interested in that stems from, this is really from the work that I proposed as a Beckman Young Investigator. It's a picture of a cell where there are carbohydrates mediating cell-cell recognition. And of course, we know that the cell is decorated with these carbohydrate chains, and my lab is really focused on trying to understand the biological roles of protein-carbohydrate interactions and then developing strategies to manipulate them. This work led us also to be interested in using these sorts of cell surface interactions to control cellular behavior. And we also have a program to really understand the role of carbohydrates in inducing the differentiation of pluripotent human embryonic stem cells and other sorts of pluripotent cells. As you heard, we are also interested in the role of carbohydrates as signals in other contexts. And then finally, we are interested in how cells build these complex carbohydrates and intervening in those processes. So I'm going to tell you a story that has to do with protein carbohydrate recognition that really came out of our work in looking at glycan biosynthesis. So we know that we need new kinds of strategies to deal with microbes, and these are 4 different kinds of microbes, so 3 bacterial species and 1 fungal species, and we are losing our ability to fight these sorts of infectious agents because of resistance to our common antimicrobial agents. And so there's been a lot of interest in developing new strategies, and there is some beautiful work going on in that area here at Illinois. But we have been interested in how our bodies physiologically recognize these microbes. Because if we could understand sort of how we deal with microbial attacks in general, that might teach us how we might use those mechanisms to combat microbes. And of course, we know that we don't just have to combat them, we have to choose who lives in us and who doesn't, because we walk around with a bunch of microbes on us. And so the current estimates are we have about 2 to 1 microbial cells to our cells, something like that. So it's been revised a little bit down from that 10 to 1 microbial cells to human cells. But we're about maybe 2 or 3 times more microbial cells. And we have mechanisms to keep those microbial cells in the proper place. So this is a picture of mouse intestine. And you can see the microbes are stained in green here. And these are the intestinal villi. And you can see that there's a 15 micrometer Newman's line. sterile zone where there are no microbes in it. And so the entities that help preserve this sterile zone are also of interest, because if we could understand better how we maintain that, we might be able to develop new strategies to combat bacteria. So, as I said, we are interested in this question. Our hypothesis is that the carbohydrates play really important roles. Now, you probably know that carbohydrates can influence how you feel, right? And so this is how most people think of carbohydrates. I'm sorry, my slide got a little messed up, but basically this person says that they're responsible for all of these degenerate conditions. This actress points out this, and this is the only statement that I think is true here. So we are less interested in this part of the carbohydrates and more interested in the carbohydrates on the surface of cells. And so what I want to do is talk about this idea that the carbohydrates are functioning in us as cellular IDs. So I'm going to assume that not a lot of people think about carbohydrates as much as I do. And so if you do, that's okay. Hopefully you'll just stand by me for a few slides. But essentially, if you look at every cell that we know about on the planet, every cell on the planet that we've studied has a carbohydrate code. So that goes from like bacteria to archaea, scremophiles, to plants. Of course, the most abundant organic molecule on this planet is cellulose, a polysaccharide, a carbohydrate, and of course, a key element of plants. And then we and our furry friends also have cell surfaces coated with carbohydrates. So these are an evolutionarily conserved feature of every cell on the planet. And when you think about what you need to have a cell, right, you need information that can be decoded, and you need proteins that can take the system out of equilibrium, that can catalyze reactions, and you need compartmentalization, right? So you can't have your genetic information just freely floating in the prebiotic soup. But it's not clear why you need a candy coat, right? But that's what every cell has. And so that is, I think, really interesting to think about what is the major role and why do we have these. And so, as I said, we are really interested in this idea that they're used to tell cellular friends from cellular foes. So here's a friend-foe picture. Yeah, everyone sees this is sperm-egg binding. And if you didn't care about carbohydrates before, I should point out to you that you began with a protein-carbohydrate interaction. So binding of this sperm to the egg is mediated by a protein on the sperm that binds to a carbohydrate on the egg. And this was really beautiful work by Ann Dell's lab. And the carbohydrate that mediates this sperm-egg binding was determined in 2011, or at least published in 2011. And that highlights that there's so much low-hanging fruit in this field. So here's a fundamental interaction, and we just figured out how it worked. And so we are, again, really interested in this area because of that. Now, when you look at cells, you might think of the proteins that are on the surface of the cells, but this is kind of what the cells look like. They have this really, sort of quite thick array of carbohydrates that sticks out from the top of them. And so if you look at a lymphocyte, you can see that if you stain for the carbohydrate layer, has quite a thick layer as well. So this is a textbook picture there from the molecular biology of the cell. And this sort of carbohydrate layer, of course, is also found on prokaryotes. So here we have Streptococcus pneumoniae, and you can see the carbohydrate layer there. And again, I'm just going to apologize. I got a new computer, and somehow the fonts are not liking me, but hopefully you'll be able to see things as we go. And so we think of this carbohydrate layer here as an ID card. And that carbohydrate layer can be used by other cells or the physiological system to really ID the cells and ask, do I want this cell or do I need to get rid of it? So let me just show you. You already know that this can occur if you know what blood type you are. So here is Lewis B, and here is Lewis A. So these are the blood group antigens. And if you're blood type B, you put one sugar on your cells. If you're blood type A, you put another sugar on your red cells. And you know that your immune system can readily distinguish between those. And in fact, there's a lot of interest in using unique carbohydrates that are on tumor cells as tumor vaccines. So these are being studied by the Wong and Danishevsky labs, as well as other labs. So you can build all the mammalian carbohydrates. So despite the variation and the structural variation, which is really quite beautiful, you can see the common building blocks, right? Fucose, fucose here, galactose here, galactose here, galactose here. So you can build all of our mammalian glycans. So by glycans, I mean glycoproteins, glycolipids, and polysaccharides. You can build them all with about 35 building blocks. And bacteria, though, are far more diverse in the carbohydrates they use. And they use very interesting deoxy sugars. They use elaborated sugars that have a wide range of functional groups. They use sugars that have phosphoglycerol. And in some cases, they use furanose sugar. So this is galactofuranose instead of galactopurinose. So in fact, to make all of the bacterial polysaccharides, you need over 700 monosaccharide building blocks. So what that means is that bacteria have enzymes that they're using to build up these cell wall saccharides that we don't have. And those enzymes are potentially targets for antimicrobial agents. And so this is my simplistic view, right? These are our building blocks, and those are bacteria. And you can really tell the difference between these two easily. And so we started thinking about that, and we've been interested in really exploiting the differences in those enzymes. And so one of the projects in the lab is to develop inhibitors that block key biosynthetic pathways for the synthesis of unique polysaccharides in bacteria. And we were especially interested in galactofuranose. So again, this is the equilibrium between galactopurinose and galactofuranose. And galactofuranose was first brought to our attention by Sam Turco's lab. So Sam works on leishmaniasis, and these sort of protozoan parasites cause diseases, not only like the skin disease, but also malaria. So galactofuranose is a critical part of the parasite's coat. Galactofuranose is also found in mycobacteria, the bacteria that cause TB, and our lab has been working on it in that organism. And then this one I won't keep up very long, but galactofuranose is also present in nematodes, and this is one of the least horrible pictures I saw of nematode infections. So it's found, galactofuranose is found in these important pathogens, and every pathogen that has galactofuranose in it has an enzyme called UGM. So you can detect, you can at least say whether or not a parasite's likely to have galactofuranose by the presence of a UGM. And UGM is just the enzyme that takes UDP galactofuranose and converts it into UDP galactofuranose. So without this enzyme, you don't have this building block, and therefore you can't make any galactofuranose containing glycans. Therefore, we've targeted this enzyme and identified small molecule inhibitors of the enzyme, and we are testing these. They show both activity against mycobacteria, but more recently, and this actually has been published just in the last few months, so they show also activity against nematodes. But the fact that there are these interesting saccharide residues led us to wonder whether there were any proteins in us that would be recognizing these unique saccharide residues that are only found in microbes. And so we thought that would be really interesting if there were proteins that could do this, because such proteins could be, for example, you could use cytotoxic agents to mediate cell killing. You could recruit the immune system to pathogens or other microbes, or you could monitor and influence the composition of the microbiota. So we decided to look at and ask the question, are there any lectins that bind galactofuranose? And so, and actually, if you ask more broadly, you know, are there proteins in us that bind microbial sugars? Here's a picture of about 40 human proteins that are lectins. They're classified as lectins on the basis either of their ability to recognize sugars or sequence homology. And these lectins in circulation, their concentrations have been measured. It's not clear what their concentrations are in these other sites, but let me just point out how little we know about these proteins. So these are evolutionarily conserved proteins, and I think my favorite is H-vicolin. So H-vicolin is a protein that looks like an innate immune protein. It looks like it should be promoting clearance of microorganisms. Its concentration in serum is about 25 micrograms per mil. That is really high. It's one of the highest concentrations for any protein in serum. And what does H-vicolin bind to? Your guess is as good as mine. So there are all these proteins that are out there that we don't know very much about what they bind to. So they're clearly a part of our innate immune system. And so we are interested in them as microbial gatekeepers. And in fact, there are already well-known examples of these. So mannose binding protein which is shown here, is a protein that has a collagen-like triple helix and 3 carbohydrate recognition domains. And you can see mannose stopped in here. These carbohydrate recognition domains have calcium in the binding site, and they bind mannose, and the protein's a nice trimer. And if you are missing this protein, you are susceptible to Staph aureus infection, P. aeruginosa infection, and children lacking this protein suffer quite extensively multiple infections. And we know something about how this protein works in that there are studies showing that it can bind to microbes via cell surface carbohydrates, and basically these coil regions here form a larger organized coil that then recruits complement and kills the microbe, but also recruits the immune system for uptake and antigen presentation. So these proteins have a really important role in innate immunity, and it's very well understood from these sorts of pictures. But what's not really understood is, we have mannose residues on some of our cells too, and many microbes have mannose residues. This protein doesn't kill all microbes. And so how does it choose who it kills and who it doesn't? And if you actually look at the list of all of these soluble innate immune lectins, mannose-binding protein recognizes mannose. Glycans, there are 14 of these. They recognize lactose. This protein recognizes lipids. This one, not so clear. This one, glycan. This one, mannose. So basically, we have all of these proteins that recognize kind of similar carbohydrates and carbohydrates that actually we put on our cell surfaces. And so why would we have, for example, multiple proteins that recognize mannose? Unless organisms that have mannose are really grave danger to us, it's not really clear. And so we think that these views are too simplistic, that we don't really understand how these work or what's going on. And I'm going to try to convince you that we know less than we think we know. But hopefully I'll tell you something new that we actually do know. Okay, so can these lectins serve as gatekeepers? So the story that I'm going to tell you starts with these guys, and then was picked up by these people. And these were a great set of collaborators. So Federico at Wisconsin, and Jim Paulson at Scripps, and Rick Cummings, then at Emory and now at Harvard. So, okay, so let me just tell you about human intellect 1 first. So we started studying this protein because if you just Google galactopyranose-binding lectin, this is the protein that comes up. That's not really how we found it, but that is true. And it is a soluble lectin that's produced at these epithelial barriers, so lung and intestine, and it's a marker of diabetes. So it's upregulated if you have diabetes. It's upregulated if you have cancers, especially gastrointestinal cancer. And there are human polymorphisms associated with Crohn's disease and asthma. So these are all kind of inflammatory conditions, and this protein is produced by cells that help maintain this barrier. And this is actually not our work. This is, again, this intestinal staining is from Laura Hooper's lab. But let me just give a picture, a schematic picture. Basically, at the bottom of these sort of intestinal villi are Paneth cells, which secrete antimicrobial peptides. And these antimicrobial peptides mediate the lysis of microbes that happen to enter the mucus. They also secrete mucus, which are highly glycosylated polymers. And we are working on making mimics of mucins in the lab. But they also secrete lectins. And of all of the molecules that they secrete, the lectins are really the ones that have molecular recognition attributes to them. And so we were interested in those attributes. And again, human intellectin is one of these lectins that's secreted. And it was identified as a galactofuranose binding lectin by Suchi et al., who showed that if you added soluble ribose or this disaccharide, which has a galactofuranose, you could block binding of intellectin to microbial cells. And you can see that these concentrations are pretty high. And so we decided to look at this lectin, and we did some preliminary experiments, but the main way we looked at its specificity is to use a glycan array. So glycan arrays are available if you have something of interest and you want to know a protein of interest and you want to know if it binds a sugar and or what sugar it binds. NIH has a facility that you can use to study its binding through again, this NIH-funded facility. So we did a study. So there are about 600 different sugars that have been just immobilized on a chip, and you just monitor where they bind. And when we looked at human intellectin, it bound to nothing. It's a great experiment. And basically, these are all human, mostly human glycans. So there were about 600, and we didn't see any binding. But there's a microbial chip, and when we tested binding to the microbial chip, again, much smaller, about 300 sugars, we saw a lot of binding. And so this, let me tell you that the number of lectins that have been run through this microbial glycan array is about 3. So these tools now allow us to examine human versus microbe binding. So it's really, really an exciting direction. But what you might wonder is all that binding due to galactofuranose? And so we ranked the top 15 hits, and then we looked at the structures. And I noticed some people have used the carbohydrate code, and the carbohydrate code is great, but you really need to draw out the structures for this. And so I'll just point out that we had hits that had galactofuranose, we had hits that had phosphoglycerol, and we had hits that had KDO or KO. And so this made no sense, because lectins typically bind a single carbohydrate epitope. And we therefore turned to structural biology to try to understand this. And I will say, I initially thought it was probably not real. But Pun in my lab, Pun Wongkanat, in collaboration with Katie Forrest's lab, was able to solve the structure of human intellectin. Human intellectin is a disulfide-linked trimer, as shown on this slide. And we were able to diffuse in allyl galactofuranose. And you can see the allyl galactofuranose here in black. And all of the allyl galactofuranose, you can see its binding is to the same base. So this looks like a protein that could sit down on a cell surface. And we were really excited that we had galactofuranose bound, because then I could figure out how this protein could recognize galactofuranose. And what those data showed is that the furanose was really just a scaffold. It wasn't recognizing this part in any detail. What it was recognizing is this terminal 1,2-diol. So the protein is a calcium-dependent lectin. You can see in green is the calcium. And it's coordinated by an array of anionic residues. And then the protein actually uses like this histidine to make this a better ligand for this calcium. And you have to have this binding to an exocyclic methylene because this CH2 group is right up against an aryl group. And so you can see nicely from this that you can't have a substituent here. So this can't bind ring oxygens. It has to bind these exocyclic, an exocyclic diol. And so this is present in every single one of those hits. So this made sense to us. And so then we asked, is this mode of binding conserved? So one thing I didn't tell you about human intellectin is intellectins aren't restricted to humans. They're found actually even in very primitive organisms. They're often referred to as X-type lectins because they were first identified in Xenopus. But they're found in every mammal that we've looked at. And they're found also in like fish, eel, a lot of other non-mammals, including sea squirts. So these are actually primitive organisms, but every single one of these organisms is a member of chordate or chordata. So you don't really think of these as having a spinal cord, and they don't. But apparently at the embryo stage, they have a little cord. And so these, all of the chordates, or virtually all of them, appear to have intellectin. And so what we did is we took the frog homologue of human intellectin and crystallized it. And we were able to get a structure of that frog version with phosphoglycerol bound. And you can see that, right, even though there's a phosphate here, that's not interacting with the calcium. We see beautiful binding that's very similar to the 1,2 diol. And this is an overlay. The frog structure is in blue, and the human structure is in wheat. And one of the things you can see is that aromatic box is really conserved, right? You have a tryptophan up here, and you have a tyrosine here in humans, and a trip here in frogs. So that feature is really conserved, suggesting it's playing a common role in both of these species. That's my joke. Okay. So I don't want you to think that this is as simplistic as I've made it sound, because it seemed really simplistic when we published our work initially, but then recently, of course, you do more experiments and you realize it's not that simple. So we have looked at binding of human intellectin to all three of these, and you can see, right, they're very related, but heptose is lacking some of the features here in KO or KDO. And when we looked at binding, and when I say we, I mean Caitlin in my lab worked with Paul Kozma's lab. And what we saw is that we see great binding to galactofuranose, to KDO, to KO, but not to LV heptose. So actually, independent of stereochemistry, we don't see any binding to heptose. So we don't understand that. So we think that this code, this microbial binding code, is more selective than it seems. And of course, a 1,2 diol is not enough. And we also know that from humans, because this is, right, the glycan in Yersinia, and that right away made me think of sialic acid. So in the human array that we tested, that we saw no binding to, there were 148 sialylated glycans, no binding to those. And Of course, we don't know why, but we could use modeling. And we see that these KDO fits really nicely in the binding site, but we could not figure out how to fit sialylated glycans. So we think that the human protein has evolved against binding our glycans. So it's evolved basically to avoid binding to sialylated glycans. Now, we don't just have one intellectin, we have two. Human intellectin 2 is also at your mucosal barriers, and it's upregulated in infection dramatically. And if you look at its binding site, so we just built this homology model, we don't have a structure yet, but based on homology, it's 85% identical to intellectin 1, so we think it's going to be very similar. The binding site is highly conserved. There's likely, it's dependent on calcium. We know that the lectin binds calcium. But there's a serine here instead of that aromatic wall. And so we began to think about the role of these aromatics in dictating the specificity. And so I'm going to take you on a little journey through physical organic chemistry now, because this is like such a great place for physical organic chemistry. So what are the drivers of protein carbohydrate recognition? And this is what I learned when I started reading about it. So bifurcated hydrogen bonds help drive protein carbohydrate recognition because you get release of water, right? That's beneficial. Calcium coordination is found in many of these carbohydrate binding proteins. And so that seems to facilitate recognition. And then something that Flo Kiocho first pointed out is that in the structures he was solving, these are periplasmic binding proteins found in carbohydrates, they had aromatic residues in the binding sites. And so these were the proposed drivers. And the origin of the aromatic residues, the argument was that these sorts of molecules are amphiphilic, right? And they have CH bonds. And these CH bonds are hydrophobic. And therefore, you want to put a hydrophobic group under this ring to interact with these hydrophobic CH bonds. So since those sorts of studies, those correlative studies, there's been a lot of data that's come out. There are a lot more crystal structures of protein-carbohydrate interactions. And so we decided to ask, what are actually the residues found in these binding sites? And so the way we did that is collaborate with Gail Bartlett and Deck Wolfson, and then Kieran Hudson was a grad student with Deck, and then Roger Deal is a grad student in my lab. And we basically did a bioinformatic analysis of the residues that are found in carbohydrate binding sites. And if you do this bioinformatic analysis, and again, we're scaling this propensity, intrinsic propensity of different amino acids found in proteins to the propensity to occur at a carbohydrate binding site. And so we have hydrogen bonding residues. There's no, some are up and some are down. There was no big increase in the propensity to have these bifurcated hydrogen bonding residues. There was actually a decrease in hydrophobic residues. So hydrophobic residues are not really found near carbohydrate binding sites, but there was a big increase in tryptophan. So tyrosine and tryptophan were the most increased. So this preference was not for hydrophobics, it's against hydrophobics. It's maybe neutral for bifurcated hydrogen bonds, but there's really a preference for electron-rich aromatics. And if you look at the preferences we saw, it's exactly the preferences that Dennis Doherty saw for cation-pi interaction. So he sees that these occur when you have, these very polarizable groups like tryptophan. So we wanted to ask, what's the molecular basis for the preference for aromatics in these carbohydrate binding sites? So the thing about protein-carbohydrate interactions is that the carbohydrates actually vary in structure and stereochemistry. So we wanted to ask, do you always see like CH-pi interactions, for example? Do you always see these when you have certain residues, or do you see them independent of what the carbohydrate stereochemistry is? And so this is just an analysis where we're looking at aromatic versus aliphatic residues on one face of glucose versus the other face. And so we see pretty equal distribution of aromatic and aliphatic. In other words, there doesn't seem to be any big preference for one kind of interaction versus the other. Whereas if you look at beta galactose, right? So we're just changing this stereocenter. Now you see an incredible preference for interaction on one face. And I think that the best way to show this is this movie. So that is an overlay of all of the structures where we have tryptophan. Okay, so actually, before DEC, and we did this analysis, it was a bit of a prediction on my part that this might occur. And the reason I thought it might occur is that if you look at how changing stereochemistry influences CH bonds, when you have an axial hydroxyl here, right, you have overlap between this CH bond and that CO sigma star. In other words, these hydrogens, the hydrogens H3, H5, and actually H4, as shown by these double bond, no bond resonance structures, are actually more electropositive. And so we postulated that maybe these CH bonds are not just interacting with these aromatics because of a hydrophobic effect. They're interacting with aromatics because the bonds are polarized and there's an electronic interaction because the aromatics are electron rich. And so the CH bonds are electron poor. And we thought we could look at this based on some really beautiful work by this Fernandez, Alonso, et al. So there have been studies where they showed because of the shielding and de-shielding zones of aromatics, you can use NMR to examine perturbations of chemical shifts. So here's the idea. If we titrate in indole, we should be able to see potential changes in chemical shifts if that indole stacks, if that carbohydrate stacks on top of the indole. So here's the data. Okay, now these are not, you're looking at these are not big perturbations of chemical shifts, but they're incredibly reproducible. And so let me just summarize the data in a simplistic way. So if we look at the magnitude of the chemical shift, perturbation, what you see is for beta-galactose, you see the largest shift perturbation occurs at H5, but we see chemical shift perturbations at H4, H3, and H1, not at these other positions. And for glucose, we see very little. One thing that was interesting is for beta-mannose, which we didn't have enough data from our bioinformatic analysis to make reasonable predictions. You see the same kind of display. And note that has an axial CO bond. So this is more electropositive, this is more electropositive, and that's more electropositive. Those are the atoms that we see those shifts on. So, and interestingly, we don't see interactions with glucose, glycan, alpha-gal. So these are residues that don't seem to engage in these CH-pi interactions. So then we did an experiment that I love because I read about this in textbooks but never done it myself. We did a linear free energy relationship, a Hammett plot for you physical organic chemists. And so we just titrated in indoles that had electron withdrawing or electron donating groups and looked at the extent of chemical shift perturbation at this H5 position. And what we saw is the greatest chemical shift perturbation when we have electron donating groups and zero chemical shift perturbation when we have electron withdrawing groups. So this really suggests that this effect is an electronic effect. And one of the cool things that this does is it explains why calcium might be in all those carbohydrate binding sites. Because if you coordinate these diols to calcium, you're going to further polarize these CH bonds so that you get more, you enhance the interaction with aromatics. So look at this case that we have in our protein. These CH bonds should be great stackers on this electron-rich aromatic. So that makes sense because I never understood why all these carbohydrate-binding proteins have calcium in them. And maybe this is a reason. And then the other thing that I'll say about this is that we think we can use this to better make inhibitors. And the way we're thinking about this, we went back and looked retrospectively. So this is a glycomimetic, really beautiful work from Viet Ernst and John Magnani. And this drug is in, or a variation of it, is in the clinic and used as an anti-inflammatory agent. And you can see what they did, right? They replaced this group with this group. And if you look at this, they replaced the glycan, right, this residue, with a cyclohexane. Well, this residue isn't going to engage in any CH-pi interactions. That should be fair game. But they actually, so the beta-galactose, they actually added an electron withdrawing group onto it, which is actually going to increase the electronic interaction with the CH-pi interaction. Now, we don't know how this binds to the receptor, but that would make sense. Okay, so, and again, so we have this structure when you eliminate this group and replace it with a serine. And I'm not going to bet money that this is the actual endogenous ligand, but we see that now galactopyranose doesn't bind well, but galactopyranose derivatives do. This is very nascent work. I think we can probably find better ligands than this. I just think they're not in any of the current glycan arrays that we've run. So we have some work to do. So I'm almost at the end. I just want to come back and show you a little bit in a few slides what we're doing to ask the larger question what these lectins do. And so one of the things we wanted to ask is, can intellectin bind selectively to different bacterial species? And we thought we would use Streptococcus pneumoniae as a good model. So Strep pneumoniae is really an important human pathogen. The carbohydrates are used for vaccines. And so we were interested in this issue because there are different serotypes. And in fact, we could identify the serotypes shown here, 8, 20, 70, and 43. And these serotypes, obviously it's the same organism in general, right? It's the same species, it's just differing by what carbohydrate it puts on its surface. So we want to see if we could distinguish between these based on the carbohydrate. And what you can see is that basically serotypes that have galactofuranose or phosphoglycerol are all bound by intellectin, but serotype lacking an intellectin ligand isn't bound. And so we started to think that, again, the lectin is trimeric and it can bind multivalently to cells. And so we started to wonder about its ability to bind commensals. So Medzitov, I think, is coming up with a really, his model for how he thinks about health is really interesting. It's really that we are constantly living with disease. We're about to be overtaken by disease, but yet we have strategies to damp it down. And so, and this is a complicated way of explaining it. Let me explain it in this way. It's a fight between the probiotics and the angry orange antibiotics. That might go over better in Madison.
It was great.
Anyway, so we can identify a bunch of commensals that have galactofuranose just based on the presence of a UGM. And so we started to look at whether this lectin that we've been studying can bind our commensals. And what we thought is, maybe it would bind the bad commensals because maybe it's recruiting the immune system to kill those bad commensals. And so here's what we found. We looked at a bunch of different commensals, and Bacteroides are not the good guys. Lactobacillus reuteri and bifidobacteria are really quote unquote good bacteria. Especially Lactobacillus reuteri, there's really not been any evidence that it taking over is problematic in any way. And intellectin binds this quote-unquote good commensal, and also bifidobacteria are what babies have to help them combat pathogens. And so that looked really interesting, so we did an experiment that, of course, everyone would do. Bought some Lifeway kefir and isolated the bacteria and asked, how much of this bacteria does intellectin bind to? And intellectin binds 80% of the species that are in this probiotic. So one possibility is that the role of this protein is to actually keep good bacteria in the gut. And that's a hypothesis we're in the process of testing. And I'm going to just quickly show you one last slide. So these studies led us to ask, what happens when a lectin competes for bacteria in a community? So the slide I skipped over was just intellectin binding to fecal bacteria. And we know it can bind a proportion of fecal bacteria. But we made a little community here of bacteria. So one experiment that we did first, because we had binding to these different commensal strains, is we picked 4 strains. So human intellectin doesn't bind to the red or the gray strain, so it doesn't bind these 2. But it does bind L. reuteri and E. fergusoni. And it binds L. reuteri better than E. fergusoni. And you can see that, so this is a flow cytometry plot, but if we overlay, so these were individual experiments, I'm just overlaying them. So we measured binding to L. reuteri, that's plotted here. We measured binding to E. fergusoni, that's overlaid, etc. And this is an overlay of what you would see if you looked at forward scatter. So that tells you what bacteria you're looking at, what bacterial species, and then human intellectin binding on this axis. Again, this is just an overlay of these data here, right? So this was, again, an individual experiment. We just plotted them on top of each other, so you can see the binding. Now, take these 4 species, mix them together, and add in the lectin. Notice how the binding here goes away. So in this mixture, human intellectin binds only to L-reuteri. No matter how much human intellectin we add, we only see binding to L-reuteri. We see no binding to E. fergusoni. Everybody to this day who's looked at lectin binding to microbes has done it in a one-on-one way, not in a community. And so this shows that these proteins can be far more selective for community binding than they are for one-on-one binding, which is really exciting because it suggests that they could be really influencing certain species over others. So that is a direction that we're also looking in. So what I hope to convince you is that this is worth looking at. That there's really interesting lectin-carbohydrate interactions that may be important for our health. And if we could understand how these work, I mean, it's not that hard to just add lectins. And I'd rather do that than have a fecal transplant. Right? And so if we can understand this, we could manipulate our microbiome in effective ways. And so, again, we started out by showing intellectin has high selectivity, and we can understand the selectivity for microbes by this 1,2-diol. And then we can understand basically how lectins are binding in general, what are the features that are important, and finally we can use this to begin to look at binding to commensals. So these are our fantastic collaborators and the researchers who did the work. So these are the people that started the project. These are the people carrying it on. I've been fortunate enough to have support for the work through especially NIGMS and NIAID. And I think all the faculty know that this is just a figurehead, right? I just sit in the big chair, and these guys do all the real work. They really drive the projects. And this is our old home, which we really loved. And I need to take a picture in our new home. But come visit me in Boston. So thanks again for the opportunity to be here. I'd be happy to answer questions.
So before we open the floor up for questions, let me just remind everybody that we'll convene in the atrium following the question and answer session for a reception at the end of this time. So let's open the floor up and Eman.
So in terms of selectivity, so you beautifully showed what happens with a single sugar and a single protein, but I can imagine that can be even enough. Think about all the interactions and both of sugar, length, range. There has to be another dimension of this playing out the selectivity.
Well, that's kind of why we're interested in looking at cell binding. And so, but one of the, I think one of the things I can say is even from that very simple, this binds like galactofuranose and phosphoglycerol, and not these other sugars, we did see selective binding to certain species or certain serotypes of strep. So I think that there's a reasonable selectivity, but as you point out, it's not the only story. And so we don't know why we bind reuteri better than we bind like Fergusoni. So we would like to try to figure that out, and we have some ideas about how to do that. But you know, so the question is it density of glycans, is it branching of glycans, or is it intrinsic affinity for a particular epitope? So I think we'll be able to figure that out. We just need to do a few million more, no, 100. Oh yeah, sure.
So you come from plant lectins, you know, you talk about the human lectin. All of plant lectins that come from the living plants. If you ingest a diet that is high in these animal proteins, you get some kind of infections or some kind of.
Yeah, so that's a really good question. So the thought has always been that plant lectins, you know, I mean, they're incredibly valuable tools for looking at our mammalian glycan. They really haven't been run through the microbial glycan array. And you would think that maybe plants might be putting out at least some of these to recognize microbes and to combat against microbes. And so I think that's really interesting to look at, and I don't think it's been examined at all. And so I feel like, you know, data like this argues that that should be done. Yeah.
I think I missed something on that last thing you talked about, where you have those 4 commensals and when you view them together, only the very responded, but there was enough of the material to take care of all of them. What's the mechanism by which the other 3 are being excluded?
We don't know. We just presume that it's hard to get so much lectin on the surface that you saturate every site. And so, we added pretty high concentration, but we didn't add like 10 molar, you know. And so, but I think, you know, in our gut, we're going to be making these things at sort of concentrations that are similar to the ones we looked at. So sort of, we even looked at like 50 micrograms per mil. So that's pretty high. And we still saw only binding to Reuteri. So that just suggests that I think they're going to be more selective, and no one's really looked at that. So maybe we can figure out what that hierarchy is. And maybe when you start to get too much of one microorganism, maybe you can switch over to binding that one. I don't know. So that's just an element that people haven't looked at. Oh my gosh, Wisconsin people. Okay, I'm going to choose Marlies.
I thought that commensal figure was really interesting, and I wonder if you have tried varying the ratios of the different bacterial strains. Is it possible to switch the selectivity?
We did do that experiment, and we didn't see switch in selectivity. Reuteri is the winner every time in that experiment. We would like to do that in a much more complex mixture, and so we just haven't done that yet. And we are trying to also, it's a little complicated, but we've done it on fecal samples, and so a really interesting thing to do would be to actually get down to the species that we bind in a human sample. That requires a lot of deep sequencing, which is really conveniently accessible at the Broad. Okay.
Yes. So I was really excited about the hydrogen to aromatic interaction. And you had a slide where you showed that the aromatic residues are much more prevalent in these carbohydrate binding sites in these lectins. Does that mean that this sort of interaction is, do you think, pretty common in all of these lectins? Like a really common way that specificity is inactive? Or do you think that there's only some of them use this mechanism?
I think it's pretty common. And one of the things that's interesting is the placement of the aromatics. So if you're binding an oligosaccharide, they'll place the aromatics under the residue that can engage in the CH-pi interactions. And what that means too is for protein design, if you want to think about enhancing like a protein carbohydrate interaction, you can think about, mutating the site such that you place residues in positions that should lead to enhances in affinity. And the other thing I want to point out, so I'm glad you asked about this, is that this isn't restricted to carbohydrates, right? Because if you have an oligoethylene glycol chain, which is really, you know, if you have like if you have a diol, that anything that would bind glycerol, right, that would bind human intellect in, you take advantage of the same effect. So people can use that in design of other kinds of ligands. I mean, I think it's a broad principle, and with these CH, you know, if you acidify CH bonds that are over aromatic rings, I think you'll enhance binding affinity. And I think
various probes or other things that want to
right. So I think this is a much more general phenomenon that really hasn't been exploited in design of inhibitors. So people have talked about it, but not so much in the electron. It's always talked about from a hydrophobic interaction. And so I really think the electronics is what's important.
And one more minor thing, have you, to test that
What are you, Sam? No.
Yeah, sorry. To test that hypothesis, if you just do a point mutant of these electron-rich aromatic groups, so maybe something that's not as much electron-rich, maybe like maybe like a phenylalanine or something that's not quite the same sort of electronics, does that change the selectivity a lot?
It decreases affinity for ligands. So I had a slide like that, but I took it out. And we've also tested putting a hydrophobic in there directly to ask, is this a hydrophobic interaction? And the hydrophobic just completely abrogates binding.
Cool.
So, yeah. Doug?
Yes. I'm curious if there's mutations in lectins that drive autoimmune disease.
So,
Break that specificity for the microbe. All
right. So that is a really cool question. There are a lot of human polymorphisms of like mannose binding protein, and it's not really clear why, and it's not, so they've been implicated in some, so mannose binding protein has been implicated in rheumatoid arthritis and some other diseases, but I don't think people have correlated, you know, what those polymorphisms do to lectin binding. Because I think also they largely use mannose at really high concentrations, and that might not be the relevant ligand. So even these long studies, so mannose binding protein, some of these were discovered like in the 1800s. I mean, they've been known for a long time. And so we may just need to go back and make sure that they recognize what we think they recognize, and then understand better how they're working. So, really good question.
I think at this point, I'd like to move the discussion to the atrium. And before we do that, first of all, 2 notes. Laura actually made it to slide 66 of 102. So there is a lot more to talk about in the atrium. Point number 1. Point number 2, let me come back to the title of her Beckman Young Investigator Award. And I would like to challenge the graduate students and postdocs. The most important thing you'll have to do is to pick the area that you're going into. And I think you can see from Laura's talk how wise she was in making the selection that she did, and where it leads to today, and how the field still has so many unanswered questions. Let's thank her once again for her.